Yes. There we go.
So good afternoon or good evening, everybody. My name is Matthew Adams. I'm the Associate Director of Graduate Admissions for the Halmos College of Halmos College of Arts and Sciences. We have merged in the past summer here, so forgive me for jumping in the old title. But I want to welcome everybody today. My name is, like I said, Matthew Adams. I do work with graduate admissions, and so does Nicole. She's our Assistant Director. She's moderating today. Nicole and I obviously help work with students on the application and enrollment process, finding the right program, from anything from the marine sciences all the way through our arts program. So if you have questions afterwards on any of our programs, we can help you out. And we will be recording today, so you'll get a chance to go back and see the slides, and God forbid, the Wi-Fi drops or whatever. But David here is an associate professor, Dr. Kerstetter, in the Department of Marine and Environmental Sciences here at Nova Southeastern University. His research interests include fisheries biology, gear conservation engineering, and fisheries policy. He's created it and taught 2 marine fisheries science courses here at NSU, and as well as 2 additional courses in fisheries policy and management and bycatch research. He has also participated in development and execution of an annual scholarship generating fishing tournament here at the Oceanographic Center in Marine Science. He has also written and contributed to various publications, including journal articles, data sets, and reports. He's also presented in a wide range of subject areas in billfish, swordfish, tunas, and commercial fishing gear technology. Today's presentation is, for the most part, it's going to be an informational webinar regarding fishery science, commercially and recreationally important coastal and pelagic fish. At this point, David, I want to turn it over to you. We're available, like I said, afterwards for questions. We do have a couple graduate programs some of you are interested in or already enrolled in, our MPS in environmental science, MS in marine science, and our Ph.D. in oceanography and marine biology. So, David, thank you again for agreeing to speak with everybody today. This is going to be an awesome opportunity for a lot of different people from NSU and across the world. So,
wonderful, Matthew. Thank you for the introduction, and thank you for setting expectations high. So, my talk today is actually a combination of a couple things. So, one, I'm going to introduce a little bit of discussion about what is fisheries, what is fisheries biology, what is fisheries science, and how those are a little bit distinct. I'll talk about what my lab does. As Matthew said, we do an awful lot with our various graduate and undergraduate students. And then I'll talk a little bit about bycatch, particularly from a fisheries perspective, the sort of issues that my lab has dealt with, frankly, since there was a lab of my own back when I was a graduate student up in Virginia. And I've carried through a lot of these same concepts through to my research program here. As Matt alluded to, there will be some opportunity for Q&A afterwards, and I've set some time aside to stick around if anybody has any particular questions. So this is actually one of my favorite photos from the last 2 years. We were doing some research with the spiny dogfish fishery up off Chatham, Massachusetts, and one of my graduate students, Jackie, that was on that trip, talks about that picture being the epitome of me in my happy place. And there's a lot to be said for that. Those that know me certainly know that I'm very, very comfortable on the back end of a commercial fishing vessel. So again, we'll talk a little bit about my lab and our work, and then we'll go into bycatch and how that all fits together. So some of you may be a little bit familiar with my background. It's a little bit unusual in the field of marine science, I feel. So I originally started out wanting to do environmental law and policy. And I was always the kid growing up in the woods and going fishing and hiking and all that sort of stuff. I'm not sure that I had clean boots the entire time I was in high school and middle school. But I was always kind of straddling the line whether I wanted to do a science major or policy, pre-law. Ended up doing a pre-law and going down to the College of William and Mary in Virginia to do their joint program in public policy. decided against going to law school and instead did their joint program with the Virginia Institute of Marine Science, where I stayed throughout my master's program, graduating 2002, and then onto my doctoral program in 2005. You can actually kind of see an evolution of me in graduate school, from me working with the scallop fishery to the longline fishery to the shark research fishery, and then down to the marlin. In addition to serving as a graduate student assistant, I also had the opportunity during my graduate program to participate in the NOAA Canals Marine Policy Fellowship Program, which is where they pluck a graduate student, usually 2 from each state with a Sea Grant program, and place them with a line agency in Washington, D.C. for a year. You're intimately involved working with full-time career professionals on some aspect of marine policy, whether it's Coast Guard or domestic environment or endangered species policy. Mine happened to be with the International Division of the National Marine Fisheries Service, where I got involved very deeply into the international fisheries for swordfish and tuna, which is actually where I ended up doing the work for my master's and Ph.D. Following graduation, I did a 1-year fellowship assistantship postdoc with VIMS, working with the pelagic longline fishery down in Brazil. I then came down to Erasmus at the University of Miami and did a joint appointment with the Southeast Fisheries Science Center, where I continued work on the Brazilian longline fishery, as well as work with the swordfish fishery here in South Florida. During the tail end of that, I started teaching courses in fisheries biology up here at NSU, established a lab in 2007, and became part of the faculty in 2013. And since 2015, I've also been working with the NSU Honors College. So it's been a very nice fit for me. South Florida is a wonderful place to do fisheries biology, because literally we are surrounded by freshwater and saltwater fisheries on a daily basis, which makes it pretty fantastic. So before I get into what my lab does, I thought I'd explain a little bit of the difference between fish biology and fisheries. And I see a couple of the panelists are actually in my current undergraduate biology of fisheries class. So you remember some of this from some of our very first lectures. And for non-specialists, these two terms are often kind of not necessarily interchangeable, but certainly conflated. And so we can talk about fish biology basically as ichthyology, or the branch of zoology that deals with fishes. So things like biomechanics, reproduction, all those sort of straight zoological questions that you could ask of vultures or spiny beetles or whatever. Fish biology would deal with that with fishes. Fisheries science, or fisheries, is a bit of a hybrid discipline that allows us to appropriately manage fisheries for human use in some sense, whether commercial, recreational, or what have you that use may be. So it is inherently a multidisciplinary field. So it includes economics and oceanography and the fish biology, as well as aspects of management, to create that integrated picture that allows us to sustainably manage fish populations. So for myself, I kind of stumbled into it. Probably one of the luckiest accidents, unless my wife is overhearing, then she definitely is the luckiest accident. But otherwise, finding fisheries science is probably the most fantastic accident of my career, because it was a way for me to use all of these disparate skills that I developed in my policy years, in my biology years, in my environmental science years, into a coherent whole. And as a result, because labs tend to follow the direction of their principal investigator, in my lab, we do a little bit of both of these. So our primary research areas fall into 4 categories. And you'll notice that the name now is now the Fisheries and Avian Ecology Laboratory, because starting in January 2020, we had our first peer review publication in bird research. So with the students that are doing the avian work, I promised a renaming, and I came through on that promise. But the bread and butter of our work has always been in two main areas. So the first is fishing gear technology, sometimes called conservation engineering. So this is where we attempt to change the fishing gears themselves in some way to reduce bycatch and make the fisheries themselves more sustainable. And we'll talk about bycatch in a minute, but the gear technology is something that is a worldwide effort in pretty much every fishery globally. So you have legions of scientists all working on their own respective fisheries to try to make those cleaner. So whenever you hear people kind of badmouth some fisheries, recognize there are a lot of us working behind the scenes to make those cleaner. The next big group of projects that we work on in my lab are fish and fisheries biology. So this is the age and growth and reproduction needed to provide the life history parameters for accurate management of fish populations. So we can't set a minimum size before knowing things like age at first reproduction or how long it takes them to get to that age of first reproduction. You know, all of these different things factor into our population models for those species that we can then use to figure out appropriate harvest levels. So in this group, also finding ways to support that management effort are things like satellite tagging, as well as trophic ecology and even endoparasite ecology. And those last two are increasingly important because a lot of the management mechanisms that we're using both domestically and even internationally are now going to an ecosystem basis. So rather than manage just swordfish, you now have to think about how to manage the entire ecosystem that that swordfish resides in. So understanding the dynamics of the small fish and the squids and all of those other organisms that contribute to ultimately that population of that trophic predator. So those two areas constitute really the bulk of what we do. That said, we've also dabbled in a number of other areas, including fishery socioeconomics, where we try to figure out how valuable some of these, particularly recreational fisheries, contribute to local economies. And then since 2010, working oftentimes in collaboration with Dr. Amy Hirons and Dr. Chris Blanar, we've been working with the coastal seabird community. And this itself came out of a project years ago. We're working on a proposal to model the entire eastern Gulf of Mexico. And I was brought in because I had expertise in the trophic ecology of these large pelagic fishes. So tunas and billfish and swordfish. Somebody else had expertise in trophic ecology of sea turtles. Somebody else had phytoplankton. So you had all of these experts on this call. And at one point I turned around and I realized and asked, where are our seabird ecologists? And there were none. And I realized at that point that there was actually a scientific opening for some really interesting work. And particularly now that we're starting to see linkages between the trophic ecology of the coastal seabirds with some of these coastal fishes. So being able to see some of the complete life cycles of parasites that inhabit both coastal fishes and wading birds like blue heron. Really kind of exciting stuff. And it's something that not a whole lot of other labs do. So it's been a really interesting thing to come into it after graduate school into a completely new field and have to learn basically a new master's program worth of information while still being a PI. So it's been challenging, but a heck of a lot of fun. So the next, I want to briefly go over some of the courses that I teach. So these are marked graduate and undergrad, as well as undergraduate honors. So at the graduate level, as Matthew said, I do a marine fishery science class where we go in exhaustive detail about things that I might mention as in a sentence here, but we'll spend a lecture on going over in that course. Things like population assessments and gear reduction bycatch reduction technologies for different gear types, aging and reproductive assessment. If you're really interested in fishery stuff, that is the course to take. I'm doing a course right now in coastal policy where I draw from that master's program in marine or in public policy. And we do things like learn how to write policy briefs and conduct policy analyses, do things like benefit cost analysis. So really useful stuff if you're interested in going and working for a state or a federal agency. At the graduate level, I also do a marine avian ecology class, basically the bird version of the marine fishery science class without all the harvesting aspects. I do a marine apex predators, where we focus on large marine animals, things like the Odontocetes whales. We do some work with alligators and crocodiles. We obviously do a lot of things with sharks as well as billfish and tunas. But all of these classes tend to be very hands-on, maybe excepting the policy one. And so I carry that philosophy forward into the undergraduate classes that I also teach at NSU, including the Sharks and Their Relatives course that I'll be teaching in the winter semester coming up, the biology fishes class that I'm teaching right now, as well as even the introductory marine biology courses for undergrads. To be honest, this year has been a little bit tougher for those undergraduate classes because of the distancing requirements. But as we get a better understanding, I hope to reintegrate those hands-on components. Did want to make a shout out, particularly this semester, to some of the undergraduates that are now working in my lab. So I've often thought and believed that one of the best trainings of a graduate student is to work hand in hand with an undergraduate student. And to help teach those techniques to somebody else makes you learn them better yourself and better understand them. So I've got Hunter up here, I've got Kat over here, doing some really hands-on stuff with both tunas and pelagic stingrays. So I know Kat's on the line today, so I want to thank her for her work and looking forward to her work in the lab next year as well. One of the things that I brought with me from my graduate program at the Virginia Institute of Marine Science, or VIMS, is that a professional marine biologist has a tripartite mission, a three-part mission as a professional. And part of that is research, part of that is education and teaching, and the third part is advisory service. So both me and my lab go out and do all sorts of outreach events. Whether it is something like the Tortuga Festival, where we've had a tent every year in Conservation Village. We actually take our research overseas in places like Turkey and France and Grenada to take our bycatch reduction techniques and equipment to help them make their fisheries cleaner. I serve on a number of different advisory bodies at the national level, including the National Highly Migratory Species Advisory Panel, as well as the ICAT Advisory Committee, where both at the national and international level, we take the science that we're finding from places like my lab and my colleagues and apply that to management at those, in some cases, international level. And there's nothing like seeing research that you did yourself with your own hands on the back of a boat being translated into international management. I can't describe how great a feeling that is. We do a lot of work with local schools, including New River Middle School, which is the Broward County Public Schools magnet school for marine science. That is a girl that has never been fishing before and seeing me holding a fish, and she's not quite sure what to think about it. We get that a lot. As Matthew mentioned, I've also served on the tournament committee and as weighmaster for the NSU Scholarship Fishing Tournament that we've had for over 10 years now. Last time we had it right up at Pier 17. It is on hold until next May, but we hope to do that as well. But all of these things provide opportunities for us to actually interact and give back to the community and the fisheries that we work with. So it's something that I really hope that the students carry forward with them. And given how many of my former graduate students now work at state or federal agencies, I think that they inherently find that part of their mission as well. So getting back to the fishery aspect of this presentation, I wanted to introduce what the concept is of a fishery, because everybody has this image of somebody fishing somewhere, usually with a rod and reel, but beyond that, it varies widely. There's no common thing that we all envision when we have a fishery. And so one of the things that I talk about is in my marine fisheries science class is that a fishery is an interlocking relationship of three components between the people, the fish, and the environment. And to properly manage that, you've got to manage all three of those components. Now, we have a number of different types of fisheries, the standard commercial and recreational ones, a little bit in the United States, but much more so elsewhere in the world. We also have what are called artisanal or subsistence fisheries, where people are fishing for essentially food. A lot of times you'll find aquaculture activities included in this definition of a fishery, because a lot of the product flows that go out commercially into the markets follow the same sort of patterns as commercial harvests do. But the real question for a fisheries scientist is, particularly one working with gear types, is how do one manage a wild capture fishery? And we usually do this through a combination of different methods. So remembering those three components, we can cover each of those in turn. So we can protect the underlying habitat and ecology that allows for fish production. So, if we want to harvest, even recreationally, a coral reef associated fish like the yellowtail snapper, we need to protect that reef. The same thing applies to estuaries or to sargassum habitats offshore. So habitat and ecology is becoming an important part of modern fishery science. We can also manage it by controlling the type or the age or size of individual fish that are harvested, so that we can ensure that there are sufficient numbers to remain in the environment for reproduction and the fastest growth and highest sustainable yield. One of the other things that we can do is control the fishing mortality rate. So this is, in simplest terms, controlling how many fish die from fishing activities. And this is where some of the work that we do with bycatch reduction comes into play. Now, in general, we tend to not think so much about our commercial fisheries or the importance of artisanal fisheries, because such a small part of our diet protein is not fish-based. But for the rest of the world, it's very different. And one of the interesting things that I found in my career working internationally is just how important fish resources are for some of these developing countries. I was at a meeting with the fisheries minister in Grenada a couple of years ago, and working with an international consortium with FAO, we were talking about these new gear technologies that would hopefully reduce how many marlin were caught in their tuna fisheries, which we kind of saw as a win-win. They could catch more tuna, not catch as many marlin, which are important for the recreational side. So we were coming at it from a very American-centric perspective. And the fisheries minister looked at us complete deadpan and said, this is all great, but I'm going to tell you right now, if anything that you do jeopardizes the ability of the Grenadian people to eat fish, I'm going to say no, because that is our top domestic responsibility. So it was just really instructive for me on how different countries view these things differently. In Florida, we have our disagreements between commercial and recreational fisheries, but these are huge industries for our state. So it's somewhere, so the last hard statistics I found were 1.2, it's probably closer to $1.5 billion for our various commercial fisheries in the state, including everything from the tuna fishery, like this boat out in Clearwater, down to the spiny lobster stone crab fishery down in the Keys, up until the mullet and king mackerel fisheries up near Canaveral. We also have a $5.7 billion plus recreational fishery. That includes freshwater and saltwater, but that is a huge deal. And there are really no other state in the United States that can quite match that recreational value. It's also worthwhile to mention this aquaculture, because oftentimes we think about something like, oh, just tilapia. And that's not true for the state of Florida. We have some other universities here in particular that are developing aquaculture for things like some of the tunas and dolphin fish. But one of the big markets for aquaculture in the state of Florida is actually for ornamental fishes. as well as bait fish, things that you wouldn't normally think of as aquaculture products. Now, in my lab, we work with both commercial, recreational, as well as some work with the aquaculture industry. So there are opportunities in my lab for work with all of those three fisheries. And just to mention it, most of the photos that I'm going to be showing are actual photos from my lab research. So we're not just showing you pretty pictures that I pulled off the internet. So this is Max Appelman right here, who was with the Atlantic States Marine Fisheries Commission for 7 years before taking a job with NOAA headquarters in Silver Spring, Maryland. But he worked with this buoy gear fishery in Clearwater. This is Jenny Fenton, who's working with the Southeast Regional Office of the National Marine Fisheries Service in St. Pete. And this is a number of our different graduate students, including Joe and Noah, both working with FWC, where we were doing tournament sampling right here in Dania Beach. So one of the things that I really wanted to talk about, though, for this was talking about bycatch, because it's one of those words that you hear about in marine science all the time, and how, you know, it's universally bad, and, you know, fisheries need to be shut down. You hear a lot of negative stuff. So I'm hopefully kind of putting it in a better framework with this discussion. And then again, I'm happy to take more questions afterwards. But when we talk about bycatch, it's the catch of non-target fish and ocean wildlife, including what's brought to port and what's discarded at sea. So if we were to fish for swordfish and catch a loggerhead sea turtle, if you're fishing for cod and you catch one of these sea stars, if you're fishing for scallops and you get some of these barn door states, all those other things that you're not trying to catch are bycatch. And globally, it's actually a pretty significant threat to a lot of marine ecosystems. And in fact, at a global level, about 40% of the total fish catch is actually bycatch. And so you can see that in this diagram over here. These are representing total catches worldwide. So you can see the bycatch in blue in comparison with the targeted catches here in green. So, I mean, it's a lot. In the United States, we tend to be better about it, in part because the federal and state governments in the United States have dedicated a lot of funding to try to solve some of these bycatch reduction problems. We do have some bad actors still in the fishery. So 9 fisheries in the United States are responsible for about half of that reported bycatch. So the point I'm trying to make here really is down here, where reducing that bycatch and bycatch mortality is a way that we can help ensure fishery sustainability. And that's one of the key things that we work with in my lab. So there are 2 main ways to reduce bycatch and the effects of bycatch. So the first one is to simply reduce bycatch based on the numbers. So you can reduce the effort, you can reduce the number of boats. That's one way to do it. You can reduce the area that people are allowed to fish in. That will also decrease the number of bycatch organisms just by numbers. You can also reduce the interactions with the bycatch species. So one of our newest projects is with so-called deep set pelagic longline gear. And you can see me here out in July on one of the boats that we're working with out of Fort Pierce. So what we're trying to do in this case is avoid interactions, particularly with sea turtles and pilot whales, both of which are known to be relatively close to the surface. So less than 150 meters is where they spend the majority of their time. So we have brought in a gear type that has been used for decades out in the Hawaii longline fishery called deep set gear, which actually sets the gear at 250 meters plus. So we use these electronic devices to record how deep the gear is fishing. And by getting below 250 meters with those hooks, we have, knock on wood, so far completely eliminated sea turtle bycatch in this experimental fishery. We've also not had a single pilot whale interact with the gear. So knock on wood, we're hoping that these results continue and allow the continued harvest of these otherwise sustainable tuna species by removing the opportunity for the interactions with the bycatch species. I've got in parentheses some bycatch or some PSAT tagging. So we'll talk about pop-off satellite archival tags. But one of the things that we do in fisheries biology is also determine the habitats. So we wouldn't have known how to set the gear deeper or how deep to set it had we not had satellite electronic tagging data showing how deep those bycatch species usually go. So it often works kind of hand in glove for reducing bycatch. So have we reduced bycatch by numbers alone in the United States? It's kind of an open question. We've certainly reduced commercial longline effort in the Florida Straits, which has reduced interactions with juvenile swordfish and sea turtles. We've reduced effort in the shrimp fishery in the Gulf of Mexico. There's some rolling areas closures in the Pacific Northwest. So by decreasing the numbers of potential interactions, yes, it kind of has worked. And so what we try to emphasize for these sort of collaborative projects is that every time they have to let a pilot whale go, not only is that a hook that could have caught a tuna that you could have sold, but it's also lost time because it takes about 20 minutes to half an hour to untangle even a live pilot whale. So if we can find ways to reduce those interactions, they can still continue to fish and make money, which is ultimately what they want to do. The other way that we can reduce those bycatch effects is by reducing bycatch mortality, reducing the number of animals that die after being caught by bycatch. So what we're focusing on here is the survivability of those organisms. So there's a strong emphasis on trying to find the least damaging way to catch these animals, as well as the least damaging ways to release them. So there are two main methods that we can do with this. So we've done some work by changing gear types or equipment. So, the fishery up here on the top, that's one of the main fishing ports in Grenada. So you can see the size of the vessels that we were working with there, where we were trying out circle hooks as a way to cause fishes like billfish to be caught in the corner of the mouth and still be alive and enable live release. We've done similar work off North Carolina with weak hooks that are designed to straighten or break when caught by something really heavy like a pilot whale, but still retain the yellowfin tuna that you can sell. We've done some work in the Bahamas with New England Aquarium, trying to develop electronic devices that essentially confuse sharks away from commercial gear. So that's always kind of fun. The other thing that we can do is changing some of the fishing techniques. So how do we best encourage live release of billfishes and swordfish? This is where we also try certain gear types like swordfish buoy gear that you can see here in our Reunion Island fishery, or green stick gear in the Gulf of Mexico. Ways that we can still enable the fishers to go fish, but not kill as many bycatch. So it's kind of a win-win for everybody. And this, in my experience, has been the process that has allowed a much more collaborative approach towards decreasing bycatch interactions and collaborative research. So one of the ways that we kind of do that second part, though, is by encouraging live release. But the question always comes up, does that process actually work? So, when I first got into bycatch reduction research, it was great in the United States because for the most part, we initially were working for inshore species, and we kind of knew that most of them, as long as they weren't too damaged, would survive. But when I got to the international level, a lot of commercial fisheries, especially outside the United States, fundamentally believe that every fish that they released, no matter how good it looked, would eventually die. And so it was actually an immoral act to release them because they would be simply wasted. So getting over that mindset has been a continuing effort as I've worked in these various fisheries over the years. And it can be challenging trying to estimate the mortality of these released fishes, because there are a lot of parameters that you have to account for. What are the capture conditions? How long have they been on the line? Where were they caught? How were they caught? What were the release conditions? What were the environmental conditions? So all of these things make these sort of projects challenging, but we've done them. And particularly, we've done a number of these starting way back with my master's program research, which is why you have the separation here, for a number of different pelagic fishes. So in this little inset diagram here, we have some of these pop-off satellite archival tags, or PSATs. And you can see how the size has gotten smaller, and you can see one here that was actually deployed on a white marlin off the mid-Atlantic. So these tags record data on temperature, light level, and depth for periods, in some cases, up to a year. For post-release survival studies, we usually do 2 weeks to 30 days. They record these data, store it internally on essentially flash drive memory, then float to the surface and transmit these data through the Argos satellite system. So it's a fantastic technology working for fishes that often move 50 to 75 kilometers a day that you will never catch again. So they transmit the data back, and we're able to use those temperature and depth data to create essentially tracks of what those fish did. And for things like these two white marlin off the Dominican Republic and Venezuela, we can actually see really interesting day-night differences in behavior. So not only can we ascertain whether they survived that capture and release experience, but we can also get interesting habitat utilization that we can then take back to other bycatch reduction efforts. So we've done a lot of this electronic work, tagging work in my lab. So back in my master's and doctoral program, we did it for blue marlin, we did it for white marlin. They all show fairly similar post-release survival rates, somewhere between 75 to 90% of these animals survive that process if they're released alive. So we've been able to take that and now incorporate that in domestic and international management for some of these large pelagics, which again is fantastic and really gratifying. So starting in 2007, when I came back, or not came back, I came up to NSU from Miami. We did similar work with sailfish. I had a project with with a graduate student, Jenny Fenton, who was looking at post-release survival of juvenile swordfish from swordfish buoy gear. I've got Britt Stavercool in this kind of goofy photo from Fort Pierce and one of the vessels we're working with that is using this technology right now. Literally, she leaves for the boat tomorrow to ascertain post-release survival estimates for juvenile swordfish like these down here in the bottom left. We've also done some really fun stuff with tags not related to post-release survival, whether it's been for blackfin tuna, pelagic stingray, white sharks, and lemon sharks. So it's really, really fun technology to work with. I know Meredith is in on this seminar, and she's working actually with some albacore data that we have from some preliminary tagging efforts. So she knows definitely firsthand what we're working with. One of the aspects that I love with our lab are not just the outreach, but the collaborations. And there are lots of opportunities for collaborative research in my lab for fisheries biology. So not only do we get specimens from these various fisheries that we then use for research, like these gumpalid heads down here that I have a graduate student now. She's extracting the otoliths from those heads and using those to provide age and growth estimators. So we do it for research. We also use it for educational purposes. So a lot of the specimens that have been donated to us over the years are now part of our teaching collection that we use for things like my intro to marine biology class or my biology of fishes class. So that's been kind of fun. We also have collaborations with recreational and charter fisheries, as well as commercial fishing boats that provide the opportunity for NSU graduate students to get out at sea and collect data or to collect specimens. So in particular, I wanted to give a shout out to the Lady Pamela sport fishing fleet just south of the NSU Oceanographic Center campus on A1A that has been providing specimens for one of my graduate students in her trophic ecology project with coastal pelagic fishes. On a larger sense, the National Marine Fisheries Service, part of NOAA, has done a wonderful job over the last 20 years dedicating funding specifically to address these bycatch problems. So particularly the Bycatch Reduction Engineering Program, or BREP, which every year gives out a couple $1 million in grant monies to various bycatch reduction efforts. So the bycatch reduction effort for the sharks in the longline fishery that I worked with New England Aquarium was part of a BREP funded project. Our project right now with NOAA Sea Grant is essentially a bycatch reduction project. So one of the fun things that I've been able to do is take that NOAA funding, help develop techniques in collaboration with these fishers, and then take those to other fisheries, both domestically and internationally, to see that overall bycatch get reduced. So lots of good stuff. On a personal level, even if you're not interested necessarily in fisheries biology, there are some things that you can do that I encourage you to do. So one of the major things that you can do as an individual consumer is choose seafood appropriately. So there are lots of different guides out there. So this one is available from download from the Monterey Bay Aquarium. Guy Harvey also puts out one. And these provide a color coding of various fisheries that you can then use to figure out what are the best choices in terms of monitoring sustainability and, yes, also bycatch. And so by your choice of choosing fishes to eat that have fewer of these bycatch or sustainability problems, those fishers will target more of those sustainable fisheries. And so you're doing your part bit by bit. I'd also encourage you to support localivore fish tastes. So instead of getting something like a Chilean sea bass that comes from one of the Southern Ocean seamounts, or orange roughy that comes from New Zealand, eat something like a Florida pompano, or one of the other locally sourced sustainable species, things like lionfish even, that would not just support local fishing communities, but also in some cases help reduce the impacts of invasive species. So again, there are some things that you can do, as well as supporting organizations and actions that encourage sustainability. So things like Oceana or the NRDC are big proponents of bycatch reduction research. So you can also support those organizations as well. So I'll leave it there. Managed to go full 40 minutes on that. Probably more than enough for everybody on a Tuesday night. But my contact information is here, as well as our social media links on Facebook, as well as Twitter. So I appreciate you all coming in. And if you all have any questions, please let me know. And I'll stick around for a little while to answer any questions y'all might have now. So thank you.
No, David, that was really super in depth there, and I appreciate you going through the full gamut of the slides there. I'm sure we have a couple questions here, and I know there's some of your own students are in here, which is awesome. But there's also some outside students that may have some additional questions.
Absolutely.
I think your lab is actually one of the better social media labs out there going for at MSU, but across the other schools that have marine sciences programs there as well. I think you guys had the stingray dissection on Instagram, one of your students did, and it was awesome. I know a ton of people contacted me about it. So we thank you on that. I do encourage the audience to follow. There is our Halmos College actually has the labs listed on there. And you can go in there and then you can follow the social media channels associated with them too. So, but Nicole, you want to open up for questions? Did you get any Q&A questions coming in?
Haven't gotten anything just yet, but like we said earlier, please feel free to drop your questions in the Q&A or in the chat. Either way, we'll see them and we'll respond to them.
So yeah, if I can just follow up with you a little bit, Matthew. Yeah, you mentioned that stingray dissection. So that was, for the audience that wasn't there, a southern stingray that washed up just south of Dania Beach Pier. So we get calls, probably once a year from lifeguards and other people who find something interesting washed up on the beach and say, hey, do you want this? And generally the answer is yes. So whether it's that stingray, you may have seen in one of the earlier slides, we had a sharp-tailed mola that washed up a couple of years ago. We've had short-fin mako and scalloped hammerheads. But oftentimes what I'll do is bring those back to the lab and we'll have a dissection day. And it's just a lot of fun, especially for me when graduate and undergraduate students are able to come into the lab and see hands-on the things that I've been talking about in lecture. Just a lot of fun.
Awesome. We may have to film one of those live next time.
Fair enough. Happy to do it.
Anybody else from the audience? I can unmute you guys too as well. Maybe want to raise their hand or. Well, we still have pretty good attendance tonight. If there's no questions at all, I guess that's it. Nicole, I think you did have a question. Oh, there's one that just popped in there. You want to read that question?
Yes.
Nicole?
Okay, so this says, quick question about PSAT tagging. What does the retrieval process look like for highly migratory species that may cover very large distances in a year?
Gotcha. Thanks, Garrett. Garrett was one of those undergrads that actually has worked with me on the past. We have a project together that we hope to submit for publication in December. So the wonderful thing about PSAT tagging is that you don't have to get the tag back. So all the memory is stored in that flash drive inside the tag and transmitted back through the Argos satellite system. So the retrieval process really is, boring as it sounds, is logging into the portal at the tag company that you work with and hitting the download button. And then that gets downloaded to your computer as a zip file, and then you unzip it and you've got either Excel or CSV data files that you can then manipulate for your own analyses. So, you know, unlike prior efforts before PSAT technology, when we either had to follow the fish for a while manually, or you had to wait for fishers to return those tags, this independent reporting mechanism really has enabled us to do a lot more work with these pelagic fishes.
Now, David, from a non-science person like myself, I wanted to ask you, now, sharing the data on these things with other universities and other researchers like yourself, do you guys collaborate within, the field?
We do. So one of the conditions for NOAA awards for about the past 5 years is that you have a window to publish the data, and then it automatically gets archived with the agency. And then agency can do with it what it wants to. And I've got a colleague down at the Southeast Fishery Science Center in Miami that is actually developing what is essentially an ecosystem behavioral model that he's trying to figure out the depth distributions of these different gear types, and then overlay the depth distributions of the swordfish and the tuna to identify how those overlaps that we can then reduce them. So, all those data do get shared and do get used well beyond what we do with our scientific publications.
That's awesome. Any other questions or anything? And if not, we can let everybody go. But
we haven't received any.
All right. Well, I think you were thorough then. I appreciate it, David.
And everybody certainly knows how to get in touch with me. Some of these attendees I'll see tomorrow.
Awesome. That's great. Well, thank you, David. I appreciate it.
Thank you all for coming.
Awesome. Have a great day, guys.
Bye.
Thank you, everyone.
Nicole, you want to stick around for just a second?
Sure. I'm going to stop the recording.